Okay, today's part 2, we're going to be talking about DNA structure. This is the good stuff. Now we're getting into the really fun, awesome, cool part of biology that we are still learning so much about. Part 1 was all about the history. Please keep in mind that we are still learning every day more stuff about DNA. Okay, so our learning targets. First, to talk about structure, we need to talk about eukaryotic and prokaryotic. What are the differences between their DNAs, or their genetic information? And then number 11, so number 1 is compare and contrast the structure of eukaryotic and prokaryotic chromosomes. 11 is know what a plasmid is and the types of organisms that have them. Okay, so here we go. First, we're going to talk about prokaryotes. So just kind of as a flashback, we have already talked about prokaryotes and eukaryotes before. Prokaryotes, they are very simple cells. They lack a true nucleus, and they lack any membrane-bound organelles like a mitochondria or Golgi apparatus. They don't have anything like that. They have this rough, let me get my pen working. Okay, they have this rough area here. Maybe I can get my pen working. Pen. There we go. Okay, apparently pen's not going to work today. They've got this rough area in the center that is kind of like all the DNA condensed. It's referred to as a nucleoid. It's not a true nucleus. Doesn't have a membrane that separates and keeps that genetic information contained in there. Okay, prokaryotic DNA is found in the cytoplasm, obviously, because they don't have a nucleus. There is one circular chromosome. This one that you see here is all kind of jumbled up, but if you could pull it out, it would be circular. Okay, consists of only DNA and plasmids. We're going to talk about plasmids in just a second. DNA in prokaryotes, it will copy itself, and then as soon as it's done copying itself, the cell is going to divide. So copy, and then it's going to divide immediately after. Alright, so what's a plasmid? A plasmid is a circular strand of DNA. Okay, well, perhaps you're thinking, well, why isn't it just a chromosome? Well, it's a much smaller, and I think I've got a picture in the next slide. Okay, it's much smaller. It's just separate. It's just a little smaller circle of DNA. Okay, so here we go. Eukaryotes. Eukaryotes have a membrane-bound nucleus. They also have membrane-bound organelles. The DNA is found only in the nucleus for eukaryotes. The eukaryotic DNA, as we learned about in cell cycle, condenses into many linear chromosomes, so that's different than prokaryotes, which are circular. Eukaryotes, their DNA, when they start to condense, they wrap themselves around histones. Those are the little proteins. They wrap themselves, start to condense themselves, and then those histones coil around each other, and then it supercoils. Eukaryotic DNA is going to make a copy of itself, going to go through replication. Then the cell is going to continue to grow. That's that G2 part of the interphase. And then mitosis is going to occur, which is going to organize chromosomes. And then everything's going to divide. The chromosome is going to divide through anaphase, and then the cell is going to divide through cytokinesis. Okay, so here's just a comparison between prokaryotic and eukaryotic cells, some of the differences that they have there. You need to be more concerned about DNA. Okay, so don't write this. You've already written this stuff down. Okay, but just to show you side-by-side comparison, prokaryotic DNA found in chromosome, whereas in eukaryotic it's found in the nucleus. DNA in prokaryotes are circular. Eukaryotic DNA is going to have many linear chromosomes, and it's going to depend on what species you're looking at and the number. Prokaryotic DNA, only DNA. It's not going to wrap around histones, those proteins like eukaryotic DNAs. Both of them are going to copy themselves. Both of them are, made out of that same stuff, which we'll get into in just a little bit. But prokaryotic DNA, once it copies itself, it's going to divide. Eukaryotic, it's going to copy, and then it's going to go through more complex steps before it actually gets to divide. Alright, so learning targets. Yay, now we get to talk about structure. Number 3, recognize the structure of a nucleotide. It's the very first thing we talk about. And then describe the chemical and structural properties of DNA. And lastly, we're only going to hit part of this one, but understand the base pairing and its role in replication. Really, we're just focusing on the base pairing. Okay, so structure of DNA. First, we need to talk about what is a nucleotide. Okay, if you remember at the beginning of the year, the unit that everyone loved was the chemistry unit, and when we talked about all the monomers for the 4 major categories when we look at biochemistry. One category was nucleic acids. It's monomers and nucleotide. Nucleotides consist of a phosphate group, a 5-carbon sugar. In DNA, it's deoxyribose. In RNA, it's going to be just ribose. Okay, and a nitrogen base. Those are the 3 parts of the nucleotide that you need to know. Okay, so there are 4 possible nitrogen bases. There's adenine and guanine. Adenine and guanine, these are purines. Purines are double circles, okay, double rings. Okay, so anytime you see 2 rings, you should know automatically that's got to be a purine. And you need to, I would just work on memorizing purines are adenine and guanine. Pyrimidines are cytosine and thymine. Cytosine and thymine, those are single ring structures. Okay, so continuing on, DNA molecule consists of 2 chains. Okay, so here's one chain. Here's another chain right here of nucleotides. See, nucleotide 1, nucleotide 2, okay, all the way down. Okay, that's what our DNA is made up out of. Okay, they're arranged in kind of a ladder configuration. See right here, the bonds here. Please note that these are hydrogen bonds. Okay. Hydrogen bonds form kind of the steps in the ladder, if you will, whereas the sugar and the phosphate form the sides of the ladder. So, arranged in a ladder configuration, alternating phosphate and sugar groups. You should see that. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. It just keeps going on and on. And the rungs are composed of paired nitrogen bases. So, this gets back to Chargaff's rules that we learned about part 1, about the complementary base pairing. Purines are always going to be with pyrimidines. Rosalind Franklin actually figured this out as she was looking at the X-ray pictures that she took of DNA and realized that a purine with a purine would be too wide and a pyrimidine with a pyrimidine would be too narrow. And so she figured out, just based on the sizing, okay, that it had to be purine and pyrimidine, which is pretty cool. The ladder coils to form a helix, and that's what you're going to see in the DNA origami that we do. Okay, so just you really need to understand that the bonds that link the nitrogen bases together, adenine and thymine, are hydrogen bonds. Hydrogen bonds, if you remember, those are our weakest bonds. Okay, that's really important because when we get to replication, which is our next part of notes, when you get to replication, those hydrogen bonds, since they're so weak, they're going to be easier to break. Okay, adenine and thymine, you also need to know those have 2 bonds, 2 hydrogen bonds. Okay, cytosine and guanine, they have 1, 2, and 3. Okay, so anytime you see 3 bonds between 2 nitrogen bases, you automatically know it's guanine and cytosine. Anytime you see 2 hydrogen bonds between 2 nitrogen bases, you know you've got to be looking at adenine and thymine. Alright, so this is going back to part of what Rosalind Franklin figured out as she was taking a look at the photos that she had taken through the X-ray crystallographer. Okay, purine and purine is going to be too wide. Pyrimidine, pyrimidine, too narrow. Purine and pyrimidine, that was consistent with the X-ray data. Okay, that's it. See you next time.